to study the brain. The aged brain is very difficult to study and to understand. The main aim of our research group is the development of new knowledge and therapeutic strategies for the treatment of neurodegenerative diseases, in particular Parkinson's and Alzheimer diseases. As you know, you are medical students. I hope. The brain is a very complex structures. It is the most complex structures we know as object of scientific knowledge. And there are several structures very interesting for us. In particular, the subcortical nuclei, in which we have some neurotransmitter systems very important for the movement, for the rewarding, for the emotions, and so on. In particular, we try to study some dopaminergic pathways. As you know, dopamine is a very important neurotransmitter involved in several diseases. Neurological diseases like Parkinson's disease and psychiatric diseases like psychosis, schizophrenia, for example, or addiction to drugs, cocaine, heroin, and other drugs, as you know. In particular, we study several of those pathways. In black here, you can see the nigrostriatal pathway. The body of neurons is located in the substantia nigra pars compacta, while the endings are located in the striatum nucleus. You can see is a very complex nucleus, and this is a so-called nigrostriatal pathway. This is a motor pathway involved in Parkinson's disease. When we don't have more dopamine in the endings, we have the bradykinesia and the akinesia, in which the movement is very poor, and we have a paralysis in patients. Another important pathway is the mesolimbic pathway, starting from the ventral tegmental area, in which we have the bodies of dopaminergic neurons. We have the projections in the prefrontal cortex in the middle part, but also in the nucleus accumbens. Another important pathway, starting from VTA, is a mesocortical pathway, very important for psychiatric disorders. When we use some drugs as antipsychiatric drugs, like haloperidol, for example, we use antidopaminergic drugs. In fact, they are antagonists of dopamine receptors. Well, this is the dopamine. Is a catecholamine, very simple molecule, but very important in our life. In particular, we can see the dopamine ending, the synapses, in which we have the synthesis of dopamine starting from tyrosine, passing through levodopa, and we have the release and the interaction of dopamine with the postsynaptic receptor. Here we can see the two pathways. The motor pathway, called the nigrostriatal pathway, responsible for Parkinson's disease, and another pathway, so-called mesocortical pathway, responsible for psychiatric disorders. You can see the prefrontal cortex. In the past, some surgery procedures like lobotomy, for example, was performed just to have a deafferentation of this lobe to reduce the symptoms of psychosis in patients. We know today that this is a very important part of the brain for the pattern recognition and very high cognitive functions of our brain. Today is not more allowed to do lobotomy, of course, but we can use drugs. And the drugs are mainly antidopaminergic for psychotic, maniac disorders, bipolar disorders in which we have a very active behavior of our patients and so on. Another small pathway is the so-called tubero-hypophyseal pathway, in which the dopaminergic pathway is linked with neurohypophysis for the release of some drugs, very important for emotions. Well, we know that during Parkinson's disease, we have very important modifications in the brain. In particular, we have the deposition of some microfibrils. They are deposition of small aggregates of proteins. In particular, for Parkinson's disease, the protein involved is called alpha-synuclein, and also the Parkinsonian patients have the apoptosis of neurons and also other cells of the brain. For example, astrocytes, they are glial cells. Well, before the anatomical damage, the histological damage, we can see a neurochemical damage. What is the neurochemical damage? We can observe early neurochemical changes in neurodegenerative diseases. In particular, we can observe changes in dopamine or glutamate. They are neurotransmitters, a reduction in dopamine and an increase in glutamate. We can observe an increase in some pro-oxidant species. They are some radicals, strange radicals we can see after what is, for example, nitric oxide and derivatives, or the decreases of some antioxidants like ascorbic acid, for example. We can observe changes in glucose and lactic acid. They are substrates or products of energetic metabolism. Then it may be very important to monitor early changes of the above parameters for try to study the early studies of neurodegenerative diseases, to try to do some early therapies to prevent Parkinson's disease. In effect, we know today that Parkinson and Alzheimer's disease they are the final chapter of a very long book. When we do the diagnosis of these diseases, they started 20, 30, 40 years before. Today, our main aim is to prevent Parkinson's disease and Alzheimer's disease, but also to try to do the diagnosis of the disease in early stages. This is very important. About the etiopathogenesis, the causes of Parkinson's disease, we have a lot of theories. The first one is related to the direct oxidation of some neurotransmitters like dopamine, and these can interact with the proteins like alpha-synuclein and other changes very important related to mitochondria, for example. They are the key points of our studies: mitochondrial dysfunctions, deposition of misfolded proteins in the brain. They are detectable as Lewy bodies in patients, and auto-oxidation of dopamine. The term of auto-oxidation is related to the self-oxidation of the molecule in the presence of oxygen. As you know, we need oxygen for our brain, for our life, but the oxygen is very dangerous because it causes a lot of problems. mainly related to reactive oxygen species, as you see later. When we have a lot of pathways, but at the end we can observe an increase in reactive oxygen species, and they are responsible of the so-called oxidative stress. What is an attack from brain derivative, from oxygen, sorry, derivatives to the brain structures. From neurochemical point of view, some oxidizable molecules up to cell membranes, up to mitochondria, and so on. The sum of the damages is the disease after a lot of years. The oxidative and nitroxidative stress is related to cellular damage. And we can see two main actors in this history. The first actor is the molecular oxygen, O2. O2 is the basis for the formation of the so-called reactive oxygen species. They are the reactive oxygen species and the free radicals. This is different. ROS are not always radicals. In effect, only the hydroxyl radical is a radical, but hydrogen peroxide is not a radical. Both are reactive oxygen species. And another source of radicals and reactive species is the nitric oxide. Nitric oxide is another gaseous molecule synthesized, produced directly in the brain by the action of a specific enzyme, a so-called nitric oxide synthase, and we have different radicals. The most important of them is the peroxynitrite radical. It is very dangerous. Then this radical is very dangerous. The first one, the hydroxyl radical. The second one is the peroxynitrite radical. Both are very dangerous and they push away the neurodegeneration. Then the take-home message is molecular oxygen and nitric oxide are necessary for metabolic processes, but they can generate ROS and RNS. Well, we need some protective factor for maintain the homeostasis of our brain. They are so-called antioxidants. The antioxidants are molecules high molecular weight like enzymes or low molecular weight like ascorbic acid, like glutathione, vitamin E, vitamin A, and so on. They are necessary for maintain under control these very dangerous molecules. Then dopamine metabolism is related to ROS production. In fact, is very important the interaction between this neurotransmitter and the reactive oxygen species in aging. In fact, during the aging, now in our brain, you are very lucky because we are very young in comparison to my brain, is very important that these reactions are in progress, but we have very efficient antioxidant systems. Well, when we have an impairment, so-called redox, reduction oxidation imbalance, it means high production of ROS and low level of antioxidant, we have a phenomenon called aging. The aging can be accelerated, and the acceleration of aging can can end in the neurodegeneration. Then the Parkinson disease, the Alzheimer's disease are basically an accelerated aging. In fact, if our life will be 120 years long, then we can have every man, woman can have the Parkinsonian symptoms, but unfortunately, some people can manifest this kind of symptoms here before. I know, for example, Parkinsonian patient, they have the Parkinson symptoms starting from 30 years. 30-year-old people has a genetic Parkinson disease related to an accelerated aging. This impairment is very important. Then the dopamine is deeply implicated in this kind of metabolism. We have some factors then accelerate this reaction. For example, metals like iron, like manganese, like copper can accelerate the production of radicals, and through the Fenton's reaction, for example, can increase the neurodegeneration, can accelerate those processes. Also, nitric oxide is very important. Nitric oxide is a very strange molecule because while we introduce oxygen by the respiration through lungs from the atmosphere, nitric oxide is synthesized, produced inside our body and produced by an enzyme called nitric oxide synthase. We have different isoenzymes. For example, we have the eNOS endothelial nitric oxide synthase, and NOS, neuronal and nitric oxide synthase, but the most dangerous is the inducible nitric oxide synthase, because this enzyme produces very high amounts of nitric oxide during neuroinflammation. In fact, this enzyme is not normally present in the brain, but it is expressed during neuroinflammation. We have then a very high production of nitric oxide starting from some amino acid like L-arginine with the production of citrulline, and nitric oxide can interact with other reactive oxygen species like anion superoxide, and the sum of those results in the peroxynitrite anion formation, and this is very dangerous, as you know. We have some protection system. For example, the superoxide dismutase is very interesting in biotransformation of this species in hydrogen peroxide, dangerous but less dangerous in comparison with peroxynitrate anion. Then we have a very complex interaction system in the brain, the potentiation by metals, the attenuation by endogenous antioxidants like GSH and so on, and the equilibrium is in our brain. Now it is very delicate equilibrium, and this can be reduced year by year during aging processes. Then the idea is from neurochemistry to the disease. In fact, the starting point is the autooxidation of dopamine. Then the loss of dopamine, we start the first symptoms of the disease, and then the death of neurons, in generally by apoptosis in the substantia nigra pars compacta, and the symptoms of the Parkinson's disease, and then some problems and different therapies. Well, another important chapter in the study of the brain is the brain bioenergetics study. In fact, we have two main theories about the production of energy. You remember that energy is stored in biological system through the ATP molecule and then used by enzymes in several pathways. Well, the classical pathway in the production of energy is the Phelan's hypothesis, in which the glucose from the blood, this is a blood capillary, through the astrocyte, this strange cell is a buffer cell, through the glucose can reach the neurons in the brain. Then the classical pathway is related to the biotransformation of glucose in pyruvate and the pyruvate in ATP through the aerobic carboxylic acid system in mitochondria through the mitochondrial respiration and production of energy. Then the final product is the lactate or carbon dioxide, for example, and the glucose is a substrate. Well, this is a classical pathway in which glucose is used by neuron as energy substrate with lactate production. The lactate production is the final catabolite, but we have another catabolite that it is the carbon dioxide. An alternative theory by Magistrati is a Swiss scientist. I met him in Sicily a few years ago, is the inverse theory in which neurons can use lactate as substrate, not more glucose. And then we have a very interesting interaction, so-called crosstalk, between two kinds of cells, astrocyte and neuron. The glucose reaches the astrocyte, okay, and it is transformed in lactate through a reaction catalyzed by lactate dehydrogenase in the presence of oxygen. This is called Hyalurobic glycolysis in comparison with the anaerobic glycolysis, because we have the oxygen, but this enzyme doesn't use oxygen. Well, this reaction is performed without the use of oxygen. Astrocytes doesn't use oxygen. The lactate is introduced in neurons and converted by another lactate dehydrogenase isoenzyme in pyruvate, and the pyruvate is the first step for the ATP production in mitochondria. As you can see, the lactate is a form of pre-digested glucose available to bioconversion to neurons for the production of ATP. Here, neurons use the available oxygen. Here, the astrocyte doesn't use oxygen. This is very interesting theory and very important also under the clinical point of view. And we have a merged hypothesis proposed by Castro and collaborators, in which we can have a balance between the two uses of glucose or lactate in the brain. In fact, normally only the glucose can be used during normal neural activities, but only during the low oxygen concentration or during high activation of neurons, lactate can be the new neural substrate for the production of energy. From our point of view, it's very important to know the key role of extracellular oxygen, glucose, lactate, but introduce another very important molecule, and this molecule is the ascorbic acid or vitamin C. This is a powerful antioxidant. We call it this molecule the final sink of reactive oxygen species in the water inside of the brain, because there is a hector exchange in the brain between glutamate and ascorbate, and ascorbate is a neuromodulator, is the real switch between the glucose metabolism, the Phelan's theory, and the lactate use as energy substrate, the magistrate theory. The switch is activated by a glutamate release. The glutamase is a neurotransmitter very important in movement. In particular, we have the cortical striatal pathway in which glutamate is released during the movement. Okay, and this is very important. This is the trigger during neuroactivation for the activation of this very complex pathway. Then, the vitamin C then is very important as neuroprotector. We don't synthesize in our body the vitamin C. Our species can produce ascorbic acid in our body, but it is very important as antioxidant. In fact, we introduce ascorbic acid through our diet. We can reach 50 micromolar in the blood to 400 micromolar in the extracellular fluid in the brain, and inside neurons, ascorbic acid can reach the incredible amount of 10 millimolar. This is 200 more concentrated than in the blood. Then we can see that ascorbic acid is concentrated inside neuron. And in this amazing review written by Heidegger in 2002, 20 years ago, we can see the real concentration of vitamin C inside the neurons. Look at this concentration, 50 micromolar versus 10 millimolar. 10,000 micromolar is 2,000 more concentrated than in the blood. Then we have very active systems, carriers, transporters, then try to concentrate the vitamin C inside the brain. Once the vitamin C is inside neuron, it can be oxidized. The oxidation of vitamin C is obtained after the interaction with some free radicals, for example, with the production of the hydroascorbic acid. This is the oxidized form of the vitamin C. The hydroascorbic acid is very similar to glucose. In fact, vitamin C is a molecule very similar to glucose. And the oxidized form is quite identical to glucose, and through the glucose transporter can be transferred inside astrocytes, and here can be reduced back to vitamin C. Usually by the use of glutathione as reducent power donor, and this completes the cycle. It is very important to know this is a very important molecule for our brain. In fact, the brain of an old people has 1/2 the concentration of ascorbic acid in comparison with young people. And the take-home message is, now we have to introduce the vitamin C in your brain, because now we can concentrate the pool inside the neurons to try to maintain the balance between this antioxidant, very powerful, and the pro-oxidant that increase during the aging. This is a key point for the preservation of the brain activity. Well, another key point of our study is sleep. Sleep is very, very important, and I'll try to explain why from our point of view. Sleep is a periodic state of physical and physic rest characterized by the complete or partial suspension of consciousness. Sleep is difficult to understand because there are so many neurochemical molecules involved that today is very difficult to understand the cycles of sleep. Molecules like norepinephrine is the main switch of the sleep, acetylcholine, serotonin, inhibitors like GABA and so on, can interact in circadian cycles for maintaining the homeostasis of neurons. Sleep is very, very important for maintaining young neurons, for maintaining young the brain. Well, this is a typical example of the monitoring of sleep. This is a track I recorded in USA, in Wisconsin, in the Wisconsin Center of Sleep, in which I spent 1 year in studying the neurochemistry of sleep. Here we can see the first state, so-called wakeness. And now we are, I hope, awake, and we can study the waves from the cortex through a system called electroencephalograms, EEG, so-called, and we can study the muscles through the EMG. They are two systems in which I study the activity of neurons, and here I study the activity of muscles. Well, the first state of sleep, wakeness, is related to high frequency waves at low intensity, very small in potential, but tonic EMG, because the muscles are moving during the wake state. The second state is called non-rapid eye movement sleep, NREM sleep. Here, the waves in the brain changes because we have the so-called slow waves, very slow in frequency, but very high in voltage, in amplitude, and we don't have signal at all from the EMG because we don't have muscular activity. This is the most important part of our sleep. This is a very deep sleep in which the consciousness is no more present in our brain. We have also the part of the sleep in which we have dreams. This is called rapid eye movement sleep, REM. The EEG is very similar to the wake people, but we don't have movements at all. In this situation, we are inside our dreams, inside our another world, inside the matrix, as you remember the movie. Our body is disconnected from our cortex, and we fly, we run, we inside our dreams, good dreams, bad dreams, but in another world. And this is very interesting because the eyes are moving. It's very complex to understand, but you have to imagine that the brain is a net of neurons connected through the synapses. Synapses are the interconnections among neurons. Imagine we have at least 80 billions of neurons in our brain. Each neuron has 1,000 of connections, so-called synapses. During the wake state or the dream state, the REM sleep, we have the connection active, and the brain is producing patterns, producing consciousness, and so on. During the sleep, the neurons start to disconnect one to the other because they need to have a rest one to one. Then the disconnection of this huge network is the loss of consciousness, and every night we have this disconnection from the brain from our body and the disconnection of the synapses in the brain. We tried to study in Wisconsin with, I think that Chiara Cirelli and Giulio Tononi, they are Italian scientists. Chiara is amazing. Giulio is the chief of the sleep center in Wisconsin and one of the most important scientists for the studies of the consciousness in the world. We try to understand the role of the noradrenaline, okay, as switch between the different states of consciousness. And we confirmed partially that norepinephrine, noradrenaline synonym, is a key neurotransmitter in the switch between the wake state and the sleep state, in particular the no-REM, the slow wave sleep. This is a very complex study. Here we try to study the neurometabolomics, the energetic state of the brain, the use of oxygen, the use of glucose, production of lactate during sleep. And this is very interesting kind of branch of neuroscience. Well, what is the key point in this kind of study? Why this is so important? Because during the no REM sleep, the slow wave sleep, the brain cleans itself. In fact, the brain try to clean the extracellular space by using a very strange system. In this animation, you can see how the system works. During the slow wave sleep, in fact, we have the vasodilation of the brain, the so-called expansion of extracellular space, and the opening of channels in astrocytes, as you can see. These channels are so-called aquaporins. They drive the water from the artery to the vein, and in this cleaning, washing of the extracellular space, they transport proteins like beta amyloid or alpha-synuclein. Remember, usually during neurodegeneration, we have a strange accumulation of misfolded proteins in the brain. Then this is very important, that only during this particular part of our sleep we have the cleaning of the brain. Then you can imagine the importance of a good sleep every night for preserving the long life of our brain and preventing the neurodegeneration. So you have to pull together these tales to reconstruct the entire picture of our approach. Well, on the contrary, chronic sleep deprivation increases the reactive oxygen species production in the brain. Chronic sleep deprivation accelerates cognitive and memory decline. And we know that we have sleep disorders a lot of years before the symptoms of Parkinson's disease or Alzheimer's disease, for example. Well, another very important chapter in the preservation of brain functions is the role of the stem cells in neurodegeneration and in Parkinson's disease in particular. Well, few years ago, more than 10 years ago, a group of scientists discovered a new pathway in the brain of monkeys, and they confirmed this neural pathway also in humankind, in human brain. This is so-called the negral subventricular zone dopaminergic pathway. We have some stem cells regions in the brain, and some dopaminergic neurons are able to reach these stem cells and induce the differentiation of stem cells in the brain. Stem cells have dopamine receptors in their membrane. Then it's very important. Then, in Parkinson's disease, we have the impairment of dopamine pathway. In Parkinson's disease, we have a reduced activation in stem cell repair in the brain, because the restoration related to stem cells is dopamine dependent. You can imagine then that in a brain in which we have dopamine problems, and the stem cells activation, proliferation for repairing the damage is related to dopamine action, then if you have the impairment of dopamine, you have the impairment of the stem cell action and the repair of the brain. This is very important because we imagine today that years and years of no repair by stem cell can reduce the plasticity of the brain to counteract the neurodegeneration in decades. And this can be a very important point for the acceleration of Parkinson's or Alzheimer's disease. We know today that, we have some neurogenic regions in the brain, in particular the subventricular zone, a region under the lateral ventricula in the brain. We have some stem cells organized in different kind of cells, type B, type C, type A. Stem cells can provide new neurons, new astrocytes, new oligodendrocytes, and so on. We can keep from this region the cells, expand in vitro the cells, and produce some structures so-called neurospheres. Neurospheres are aggregates of stem cells, neural stem cells, and we can differentiate these cells in neurons, astrocytes, or oligodendrocytes. And this is very, very interesting. Some picture from some neurospheres from our lab. This is a neurosphere. We have hundreds of cells. This is a SEM microscope, and this is a fluorescent microscope. And you can see this structure, and we can see different kinds of cells. We can identify by some markers: B cells, C cells, A and E cells, and so on. The marker for stem cells in the brain is the nestin. DAPI is for the coloring the nucleus. And we have, for example, the beta-3 tubulin is a marker for neurons. This is a neuroblast. For example, a GFAP is a marker for astrocytes, for example. Then inside the neurosphere, we have a microbrain in pre-organized. We can study then the neurochemical properties of these aggregates just to understand the potential role of stem cell in the prevention or in the therapy of Parkinson's disease and other neurodegenerative diseases. Well, we can see this is a neuron, okay, and inside this neuron we can see the vitamin C transporter. If you remember, the ascorbic acid it is very important in the preservation of the antioxidant properties. Then today we know that neural stem cells have ascorbic acid inside because they have the transporter. In the other cells, similar to astrocytes, we have, for example, the transporter for glucose. If you remember the slide, we can see that this sphere has all the cells necessary for recycling vitamin C inside. Then few experiments to reproduce this kind of micro system in vitro through a aggregate system of cells called a neurosphere. Well, I show you some experiment in which the neurospheres are able to store ascorbic acid, for example. The neurospheres are able to regenerate the reduced form of ascorbate from the oxidized form. Well, neurospheres are able to protect dopamine from autooxidation. Some ideas, because it's very tough to understand this kind of experiments. Well, at the end, stem cells express increased antioxidant mechanism able to counteract oxidative stress in the brain. Today, we can see that now in our brains, stem cells are acting as a powerful antioxidant system repairing brain damage. They are repairing brain damage by increasing antioxidants, by modifying the micro system in which they try to counteract the effect of pro-oxidants and slow down the effect of ROS and RNS. Now, after this very wide introduction to neurochemistry, several aspects, I try to show you how we try to measure in real time neurochemicals in the brain through a new technology related to some neurosensors, because our aim in my lab is to try to quantify in real time the amount of important neurochemicals related to some neurodegenerative diseases. Well, this is the first microsensor I show you. This is an implantable microsensor for the real-time detection of ascorbic acid. We make those sensors in our lab. We implant in animals, now also in several kinds of mice, rats, insects, for example, for the study of the central complex. Central complex is a very old piece of brain related to basal ganglia of vertebrates, and we can implant those sensors in the brain and read in real time the amount of ascorbic acid and the changes in the ascorbic acid in the brain of awake or sleeping animal without anesthesia. This is a typical tracking of an ascorbic acid in the brain of a rat. We published this a few years ago, and this is, for example, an injection of ascorbic acid tracked by a amperometric sensor implanted in the striatum. This is the caudate putamen inside the brain. If you remember, the striatum is the nucleus in which we have the endings of the dopaminergic neurons from the substantia nigra. Another sensor, very important, is the oxygen microsensor, because oxygen, as you know, is very important for the brain respiration, but also for the production of reactive oxygen species. Here we can read the oxygen. This is a microsensor, very complex. This is a carbon tip covered with a lipophilic membrane called the nitrocellulose, and we can reach and monitor the brain. This is the first wireless monitoring of brain oxygen by a telemetric system implanted in the rat brain, which here we can see the complexity of this kind of experiments. Here, just to have an idea, imagine to have a freely moving animal. This is wake, okay? This is a telemetric small box with small electronics inside and the sensors implanted in specific brain areas. And here you have the exposure to the animal to 100% pure oxygen. You can see the increase of the signal in real time, okay? Immediately the oxygen increases after the exposure to the animal to pure oxygen, just putting inside the bowl pure oxygen. Here we put pure nitrogen, and you can see the ischemic effect of nitrogen in the brain. Here we study several drugs, for example, Diamox and other inhibitors of enzymes, and we confirmed that the system work very well. Well, this is a neurosurgery in which we implanted the first prototype. Now we have very complex prototypes, and this is the sensor we made for real-time detection of oxygen. This is the first oxygen implanted in the brain of an animal and monitored in real time through a telemetric system. We reached this target in 2009 and started our neurotelemetry lab. We have other sensors. For example, the Dopamine Microsensor is a carbon-based microsensor covered with a very strange membrane called nepheon. This is a cationic resin exchange polymer in which we can concentrate dopamine on the surface of the carbon. Dopamine in physiological condition at pH 7.4 at 37 degrees Celsius, etc., is positively charged. Nepheon is a membrane negatively charged. Then the nepheon attracts dopamine on carbon surface, and on carbon we can oxidize dopamine. This is a typical calibration we obtain in the brain with dopamine. We studied other system for dopamine detection with some Romanian scientists, and here can see the ultra miniaturized microsensor for dopamine detection in real time through a very complex technique called the fast scan cyclic voltammetry. Well, we can oxidize and reduce the dopamine on carbon surface. This is very small. Remember that one red cell has a diameter of 7 micrometers. This sensor has a diameter of 30 micrometers. Then it's very small. We can implant this very close to the endings of the dopaminergic neurons and read the dopamine in real time through two reactions, the oxidation and the reduction of dopamine. This is a typical tracking of the dopamine oxidation and the reduction, and this is the transfer of electrons from dopamine to dopamine orthoquinone, and here the back through dopamine orthoquinone to dopamine reduction. This is chemistry. I'm sorry, but we don't have the time to explain, but if you have the curiosity, we can ask me. If you want to visit my lab, I'm very happy to show you some techniques because they are very amazing. Read the neurotransmitter in real time during the behavior of the animal is incredible from a scientific point of view. We can see the release of dopamine. This is the raw original signal. Look at this on the mouse pointer. We have the baseline. Here we have a small wave here and here under. If we subtract the background signal, we have two signals. Here we have the background, and this is the peak related to release of dopamine. Every scan is performed every 10 milliseconds. Then we have the real-time release. We can monitor the release of dopamine real-time on dopaminergic index in the brain. You can see a baseline here. Look, this is very flat, and here you can see a release. Look at this baseline here, and this is the release of dopamine. The relationship between the oxidation peak and the reduction peak of dopamine on carbon surface is so-called the chemical fingerprint of dopamine in the brain on carbon surface. Okay, this is dopamine transient, dopamine release. Well, we can see over time this release, for example, after some physiological stimulation, like a tail pinch. We pinch the tail of the rat, we can see the release of dopamine related to glutamatergic and indirect activation. Okay, and other experiment with drugs like dextroamphetamine and so on. Important is another neuromodulator, nitric oxide, as you remember, is vasodilation, dilatation very important in the brain. And here the sensor is very complex. I show you some basic sensors. And this is a new sensor we developed in Ireland with the Robert O'Neill group in Dublin. This is a very interesting microsensor for the detection in real time of nitric oxide in the brain. Another very important field of neurosensor for studying the early stages of neurodegeneration is the development of amperometric biosensors. Okay, this is the huge field of biosensor. What are biosensors? Well, biosensors are analytical devices incorporating biological materials, for example, enzymes. We have a neurochemical, we have a biological molecule, an enzyme, an antibody, an entire cell, and we can read the interaction between enzyme and your molecule of interest through the transducer, the signal transducer. In our case, we have electrochemical transducer with the production of electric signals. In our case, amperometric signal, small currents. Well, this is our typical biosensor. The enzyme immobilized on the surface of a platinum sensor is an oxidase. An oxidase enzyme can transform a substrate in a product in the presence of oxygen with a byproduction of hydrogen peroxide. The hydrogen peroxide can be catalytically oxidized on platinum surface after the application of oxidation potential of plus 700 millivolts versus a silver chloride reference electrode. The small current produced is proportional to the amount of substrate present in the extracellular space of the brain. Well, as you remember, the kinetic related to enzymes is the Michael-Menten kinetic, in which we can identify the Km, the Michaelis constant related to the affinity of the enzyme to the substrate, and the Vmax is the maximum velocity of the reaction, okay, related to the amount of enzyme present in our biosensor. The enzyme is very complex, and we can study with biosensor a lot of interactions, inhibitions, for example, competitive, non-competitive, uncompetitive, mixed, ping-pong inhibition, a lot of studies we can do. But if you implant in the brain a biosensor having this design, by changing the enzyme, you can change the substrate you can read. I show you some examples. For example, glutamate biosensor. In this case, the enzyme is called glutamate oxidase. It transforms the glutamate in hydrogen peroxide. The glutamate has a very low concentration in the brain. During a release, we can reach 10 micromolar at maximum. Then we can read a small transient in the glutamate. It is very important to protect the biosensor. This is a small cylinder, a platinum cylinder. In this case, we have 1 millimeter cylinder covered with different layers in which we can have glutamate oxidase, the enzyme, the biological element, some enzyme stabilizer like the polyethylenimine, and some polymers for covering the entire design. Okay, this is our idea of biosensor. Okay, a schematic representation of an amperometric glutamate biosensor, in which we have the enzyme stabilizer, the polymer, and the idea is the substrate can reach the enzyme produces hydrogen peroxide, the hydrogen peroxide is oxidized by the platinum. The interferences, usually organic acids, like ascorbic acid, for example, or derivatives of catecholamines, DOPAC, HVA, and other molecules, uric acid, can be read on platinum surface. But this polymer, called the perm-selective polymer, allows only the passage of the substrate, but not of the interferences. Okay, here we have the extracellular space. This is a typical calibration, and changing the enzyme, you change the substrate. This is a typical example, glucose biosensor, a second biosensor. Same idea, different enzyme. The enzyme here is the glucose oxidase, for example. We can have the lactate biosensor. This design is very complex because lactate enzyme and lactate in the brain has very high concentration in comparison with glucose, for example, or glutamate. And the idea is quite the same. Okay, we can modify some enzymatic properties by acting on nets on the surface. This is an example of SEM, surface electron microscope. Here we have the platinum. In this interface we have the enzyme. The enzyme layer is a nanometer, 20, 50 nanometer layer of enzyme. Indeed, we have a huge molecule of polyurethane. When we add this huge sponge on the surface of the sensor, 7 micrometers is not so huge, but the effect on the enzyme kinetic is very interesting. During the research and development of this kind of micro devices, implantable devices, we try to modify every aspect of the sensor for reaching the optimal analytical performance related to the concentration of lactate, in this case present in the brain. For example, while glutamate has a concentration of 10 micromolar, glucose has a concentration of 500 micromolar. Lactase has a huge concentration, more than 1 millimolar. Then is 1000, 1000 times higher than glutamate. Then we have to change the design of the implantable sensor in relationship with the analyte to detect. Another neurotransmitter, this is very complex, like a book of biochemistry on the surface of sensor. Here we use two enzymes, acetylcholine esterase and choline oxidase, for the transformation of acetylcholine to hydrogen peroxide through two reactions. It's very complex, but we can use this biosensor not only for the detection of acetylcholine, but also for the detection of some mycotoxin, in this case, through the inhibition of an enzyme. In this case, we have the one aflatoxin, is a contaminant present in milk, for example, in flavours and so on. And we can detect also contaminants in foods through this biosensor. Okay. Well, another very interesting biosensor is the alcohol biosensor. And this is, we developed this sensor for the first time in the world a few years ago through the detection or real-time detection of ethanol in the brain. Okay, this is the original paper in which we published. Look at this, the several design of biosensor, very complex, several mechanistic curves, okay, but at the end, the design number 8 is the best performing design for our parameters and for the detection of glutamate inside the brain. You know the effect of ethanol, sorry. You know the effect of ethanol in the human behavior. This effect is mainly related to the release of dopamine, but few people quantified the real ethanol in the brain regions responsible for the behaviors of human. We implanted this in rats and demonstrating the pharmacokinetic of ethanol in the brain in real time. And we can study now the addiction to ethanol, the effect of anti-ethanol drugs, and so on. This is very, very interesting. Well, this is a very interesting, also only for chemists and related to the development of biosensors. When you have interferences in the brain, when we want to read some molecules inside the brain, imagine to implant a small sensor inside the brain, is like to put an entire laboratory inside the brain, because you monitor, for example, glutamate in real time, but it's very difficult to separate, distinguish between glutamate and other molecules acting as interference. Then we need to develop some kinds of polymers, so-called perm-selective films. Perm-selective films allow the passage through its surface of analytes, but is completely opaque to interference, transparent to our molecule, but opaque to interference. Then the idea is to develop some new devices for monitoring biological molecules in real time. Well, we have some monomers. We can combine them for like Lego. Okay, you know the Lego bricks. We make together one-to-one, we make a huge polymer on the surface of the sensor on top of the enzyme layer, and we can prevent the penetration of interference in the lower layers of the biosensor at nano scale level, obviously. Some experiment, and this is a typical sensor, implantable sensor under microscope. Here you can see this small foil is the polymer. Look at this, exfoliating part is a polymer, is a micrometer level of thickness, but is protective for the layers they are under. Okay, this is another picture of the surface of the sensor, and there are some sensors with some pictures. Here you can see some moon land like pictures in the surface of the sensor with the enzyme. These clamps are related to the enzyme proteins on the surface of the sensors, and here we have the transformation of glutamate in hydrogen peroxide, for example, in real time. We developed also other kind of foils for the surface using natural molecules, for example, magnolol or eugenol. They are present in some plants, and we can use this kind of chemistry for making the surface of our sensor. Look at these. They are very strange, but they have a very strange behavior also in relationship with neurotransmitters, in this case with acetylcholine. Well, the combination of this device can allow us to have a new vision of the brain from inside the brain, just monitoring the neurochemistry or the biochemistry of the brain in real time. For example, I show you some example of monitoring of the bioenergetics of the brain in real time. As you remember, ascorbic acid, oxygen, lactate, glucose, we studied the theoretical properties in the brain. Now we can monitor them together by implanting some sensors in the brain. Okay? We, through the stereotactic surgery, this is a prototype, through specific brain coordinates. This is the coordinate of the striatum of the rat. The striatum is the region in which we can follow the release of dopamine and the interaction with the cortex, for example, for the motion extrapyramidal pathway of motor pathways. Look at this. This is a tail pinch, and this is the ascorbic acid monitored by the after the implantation of ascorbic acid microsensor. Ascorbic acid, for example, after neuroactivation, we have an increase. If you remember, ascorbic acid is connected to glutamate. After we can observe a release of glutamate, we have an increase in ascorbate. This is like a shadow of glutamate. It is the glucose. During neural activation, we have a decrease in glucose, like an increase in the use of glucose as a substrate. This seems to confirm in vivo the theory of Marianne Fillens, for example. We have an increase in lactate, similar to ascorbate. And we have also an increase, because this is a negative current, of oxygen. Okay, this is an increase. During neuroactivation, you increase also respiration and the release of oxygen by hemoglobin in the extracellular space. Another important part is the multiparametric monitoring through biotelemetry units connected to multisensors. Well, we developed also the electronics. For us, the electronics is interconnected to sensors. The sensors are connected to chemistry. The chemistry is connected to neurochemicals. We have to understand every step of this complex system if you want to study every part of this complex approach with a good chance to have good results. And here we have some implantable biotelemetry units, very simple to connect to your PC. We use usually a USB. This is the receiver. This is the transmitter connected to a potentiostat two-channel system, and the weight is not so high. Okay. We have also the possibility to monitor the movement, the microvibration of the animal, for example, through some specific sensors. This is a gold sphere inside a gold cylinder. The movement, the micro movements of the sphere open and close a switch, and we can see by amplifying this signal if the animal is moving or not. Okay, and this is a very complex study about the interaction of bioenergetics with the movement of the animal. In this study, we monitored the glucose, lactate, and the movement of the animal through a miniaturized system and two biosensors implanted in the striatum. This is very interesting because, as you know, we have the bioenergetic and also the movement of the animal. And here we can see in real time the movement of the animal and the neurochemistry. For example, look at this. We have during spontaneous movement, here we have a spontaneous movement. This in gray is the motion detection, okay, the microvibration detection on the head of the animal. Increase in movement correspond to a decrease in glucose, increase in lactate, okay? Movement, glucose decreases, lactate increases. Here, movement, glucose decreases, lactate increases. This seems then during movement, glucose is used as substrate, energy substrate, and lactate is produced back after the use of glucose. This seems to be true. And it's very interesting because the synchronization of the signals is very interesting because this is the first time we can read this kind of biochemistry in real time. During tail pinch, we can see a similar effect. Tail pinch increases the movement of the animal, decreasing glucose and increase in lactate. Restraining is the restraining of the animal produces some stress in the animal, and we can see strange behaviors in which we have a short-lasting decrease of glucose, an increase of glucose, long-lasting, and an increase in lactate. This should be due to the release of norepinephrine in the striatum and the glycolysis of glycogen from astrocytes. This is a very complex pathway that we don't have the time to explain in particular. This is a drug we can administer. The dextroamphetamine, for example, is a drug that releases dopamine. After dextroamphetamine administration, we can have a short-lasting decrease in glucose, a short-lasting increase in lactate, and after this, a long-lasting increase in glucose. This seems to be also related to glycogenolysis from astrocytes. Well, we have also some neurotoxins that can accelerate the brain neurodegeneration. One in particular is called MPTP. It can intoxicate, in particular, dopaminergic neurons by killing the mitochondria just in this kind of neurons. Also, in this case, we can see the increase in glucose, the increase in oxygen and the increase in lactate. This is an increase because the cycling currents is inverted, okay? It's very complex to understand, but trust me, when you have an increase in a negative current related to oxygen, corresponds to an increase. Then, during MPTP neurotoxin intoxication, this neurotoxin produces Parkinson's disease in animals, in rats in this case. We can observe this kind of trend. Another important technique we can use, I show you some principle, but in very short time today, is called in vivo microdialysis. This device is like an artificial microcapillary implanted in the brain. This is a microfluidic system in which we can administer an artificial extracellular fluid through the microdialysis, the dialysis of the surface of the artificial capillary. We can produce exchanges of low molecular weight chemicals, and we can extract molecules from the brain and put drugs inside just to study this small neurochemical interaction in a small area. Well, this system a little bit more complex in comparison with the sensor. We need the pumps, the fluids, and we implant this kind of semi-permeable membrane in the brain, and the fluid recovered, called dialysate, is analyzed by HPLC with electrochemical detection or other fluorimetric analytical system for the detection of, for example, neurotransmitters and bioenergetics molecules. Okay, MPTP is a molecule, neurotoxin, that induces Parkinson's disease in animals. We can study the effect through in vivo microdialysis of MPTP on glucose. For example, glucose decreases after the neurotoxin effect after 3 days. The pyruvate decreases, but lactate increases. Then we have the switch after mitochondrial damage, the death of mitochondria, through the glycolysis, anaerobic glycolysis, because mitochondria are not more effective in the production of ATP. And then we have apoptosis of neuron, no more able to produce high amounts of ATP. Okay, now we try to define the multiple roles of bioantioxidants in the brain. Well, what is our idea? That during our life, we introduce a large amount of antioxidants in our body through food. And they are introduced mainly from vegetables and colored fruits on other vegetable, in which the color is representative of a different family of chemicals. You know, the entire family called polyphenols. Okay, polyphenols are molecules that are very important for the brain. Then we have vitamin C in fruit. Then we have vitamin E, vitamin A, but also polyphenols, small molecules produced in plants, in fruits in particular, that are able to protect the brain from oxidative stress. We have a lot of bioantioxidant, bio because they are contained inside fruits. One long life of introduction of antioxidants in the brain is very important for preserving brain activity and slow down the aging of our brain. This is the idea. One of the most studied molecules is the polyphenol called chlorogenic acid, contained in a lot of red, blue, red fruit, black fruits, for example. Because remember, the color usually is related to the content of polyphenols. For example, you know the grapes, okay? The grape, the black or red grape from which we produce the red wine is fully contains a large amount of polyphenols. The red grape has a lot of polyphenols in the external part of the the grape. The white grape contains very few amounts of polyphenols. Then we know today that the red wine is more antioxidant than the white wine because the color is representative of families of polyphenols that they have anti-aging, anti-cancer, antioxidants, anti-inflammatory effects. And then we know today that we have a lot of effects mainly against the neurodegeneration. Well, we know, in fact, today, and we studied in the past, that in the brain we have an homeostasis among antioxidants in the water phase, in the lipid phase, endogenous antioxidants, exogenous antioxidants, and we introduce antioxidants mainly through our diet. Then it's very important today, if you want to have a long and healthy life, to select the food to introduce in our body for protecting our brain. This is a key point in our diet. For example, we know that in India, they suffer less of Parkinson's disease in relationship with our West society, Western countries, so-called. Why? They use some spiced food like curcumin, like ginger, for example, and the spices contain some antioxidant that exert neuroprotective effects. One of the most studied molecule is called curcumin, and curcumin has a very anti-aging properties. This is very, very important. Then, in our study, we can try to study the properties of antioxidants in food and in plants. Okay? For example, vitamin C is contained in orange juice. Then, if we study the vitamin C in orange juice and we study the vitamin C in the brain, we can transfer the neuroprotective effect from juice, from orange to juice, and to juice to brain, if we preserve this. And this is very important. And we make also sensors for monitoring antioxidants in foods. This is a very interesting approach. I show you some interesting results we discovered in fruit chopped and a salad in the market, for example. And we selected kiwi, pineapple, and melon. They are very rich in vitamin C, but the amount of vitamin C, remember that when you have a molecule inside a food, and this molecule has a healthy effect like a drug, we call this molecule nutraceutical. Nutraceutical is a drug inside a food. Okay? Then, in our case, the nutraceutical is the ascorbic acid or vitamin C. Well, day by day in chopped fruit, we can observe a decrease in vitamin C. Then, if you put the chopped fruit on Monday, on Friday, we don't have inside the fruit a good amount of vitamin C. And the vitamin C, as you remember, is very important for our brain. Then, in this interesting paper, we published in Food Chemistry, an important journal about healthy properties of foods. We demonstrated that if you have your chopped fruit at a very high temperature, for example, or you stop the low temperature and reintroduce the low temperature after a small amount of time. For example, the temperature are very, very high here in Sardinia in summertime. Then in summertime, we don't have large amounts of vitamin C inside our foods. The take-home message is fresh fruit is good. Chopped fresh fruit is good on Monday, but on Friday the vitamin C amount can be non sufficient for transferring the nutraceutical properties to our biological system and finally to our brain. This is very important. The fruit seems to be the same, but the content is completely different, and we are interested to the healthy content of our food. Okay, another biosensor for studying the same, very complex in this case, but the same idea, monitoring the healthiness inside food for our brain. Okay, and here we study a different blueberry, in this case kiwi, and so on, some papers just to see. Okay, another monitoring of ascorbic acid inside another plant. They use Mexican people used to eat this plant, and it is very rich in vitamin C. But the presence of vitamin C is related to the water you administer to the plant. Then it's very important the equilibrium, the ecosystem around the plant, around the food, around the brain. Well, here we can study also in vitro the properties of several fruits phytochemicals. For example, we selected blueberry and melograne. They are two interesting fruits, very, very rich in sugar, but very, very rich in polyphenols. And we demonstrate that the polyphenols inside the fruits are able to stop the intestinal absorption of sugars, glucose in particular. Then, very sweet fruits are don't have the glycemic peak as white sugar, for example, we use in our house for making tea, coffee, and so on, because the polyphenol present protect our body from glycemic peaks, then can protect our brain from glucose peak. If you remember, the ascorbic acid is recycled by glucose transporter. If you introduce in your brain a glucose peak, the glucose can do a competition to the transporter with the ascorbic acid, and this competition can interfere with the antioxidants properties of ascorbic acid. Then the idea is more high glucose in the brain, a peak, less antioxidant properties of ascorbic acid because of the competition with the transporter. Then natural fruits have inside polyphenols that are hypoglycemizant. Reduce the peak of glucose in the blood and then in the brain, because they stop, for example, the transporters. They block the transporter of glucose at intestinal level. And this is another very interesting point. Another source of antioxidants is, for example, the oil. In our tradition is the olive oil, extra virgin olive. Is fully of oil, we can reach 16-17% of weight in vitamin C, a huge amount. And in some very important journals about medicine, in particular the New England Journal of Medicine, the Mediterranean diet in which the olive oil is a key component, has a neuroprotective effect and also preventive in cardiovascular disease. But we can reach also the beer because recently also the beer is very interesting, similar to red wine. You remember, red wine has been studied for a long time because it seems to have antioxidant effect and so on. Recently also beer is very interesting for us because the polyphenols contained in the beer can have neuroprotective effects, okay? Ethanol is not so good, but polyphenols are very, very good for the brain. Then we are very interested also to study this kind of production also for brain health. Okay, for today we stop here our speaking.